In the eastern reaches of France, where the Jura mountains rise like ancient sentinels above valleys thick with vineyard mist, a boy was born into a world that did not yet know its smallest inhabitants. The year was 1822. The month was December, when frost silvered the cobblestones of Dole, and breath hung visible in the cold air, visible, while so much else remained invisible, unknown, unnamed. Jean-Joseph Pasteur, a tanner by trade, welcomed his son Louis into a household that smelled of leather and labor, of the chemicals that transformed animal hides into useful goods. The mother, Jeanne Etiennette, held her infant close, unaware that this child would one day transform the world as thoroughly as her husband transformed raw skins into finished leather. The tannery was a place of transformation. Animal hides arrived stiff and useless, passed through vats of lime and bark extracts and mysterious preparations, emerged as supple leather that could be shaped into shoes and belts and saddles. The process required knowledge passed from father to son, required patience and timing and attention to subtle signs that distinguished success from failure. Sometimes the hides emerged perfect. Sometimes they rotted into worthless ruin. No one could fully explain why. The variables seemed random. Temperature, perhaps, or the quality of the water, or the phase of the moon for all anyone knew. The tanner worked by tradition and intuition, by accumulated wisdom that could not quite articulate its own foundations. Young Louis watched his father's careful attention to process, watched the transformations that seemed almost magical, watched the failures that came without explanation. The boy was quiet, was serious, was more interested in drawing than in academic subjects during his early schooling. His talent for portraiture was genuine. His pastel drawings of family and neighbors showed real artistic ability, showed capacity for patient observation, for capturing what the eye perceived. Had circumstances differed, had certain teachers not recognized something else in the boy, Louis Pasteur might have become a provincial artist, might have spent his life rendering visible the faces of his neighbors, rather than rendering visible the invisible world that surrounded them all. But something shifted. A teacher named Romanet saw potential beyond artistic talent, encouraged the boy toward academic pursuits, convinced his father that Louis deserved education beyond what Dole could offer. The family moved to Arbois, a town slightly larger, with a school slightly better, where the young Pasteur began to show the intellectual intensity that would characterize his later work. He was not a prodigy. His early academic record was competent but not exceptional. What distinguished him was persistence, was the refusal to accept incomplete understanding, was the need to pursue questions until they yielded answers. The Jura region shaped him. The mountains and valleys, the vineyards and forests, the seasonal rhythms of agricultural life, all impressed upon him the importance of natural processes, of transformations that followed laws even when those laws were not understood. The winemakers of the region performed their annual alchemy, turning grapes into wine through fermentation processes they controlled through tradition rather than science. Sometimes the wine was excellent. Sometimes it soured into vinegar. The variability seemed inherent, seemed beyond human understanding, seemed simply the nature of things. But young Louis absorbed these observations, stored them in memory, would return to them decades later when the questions finally found their answers. The religious culture of the region also shaped him. Catholic France took faith seriously, and the Pasteur family practiced their religion with quiet devotion. Louis would maintain this faith throughout his life, would see no contradiction between scientific investigation and religious belief, would view his discoveries as revealing the glory of creation rather than diminishing it. The invisible world he would later discover was, for him, evidence of divine craftsmanship, complexity beyond human imagining, order underlying apparent chaos, purpose woven into the fabric of existence at scales too small for eyes unaided by lenses. At 18, Pasteur traveled to Paris to prepare for entrance examinations to the École Normale Supérieure, the elite institution that trained France's teachers and intellectuals. He passed the examinations but ranked 15th, acceptable but not distinguished. Dissatisfied with this mediocre showing, he withdrew, returned to his studies, took the examinations again the following year. This time he ranked 4th. The persistence was characteristic. The refusal to accept less than excellence was already evident. The boy from the tanner's workshop would not be content with competent. He would pursue mastery. The Paris he entered was a city of intellectual ferment, of scientific societies and public lectures and laboratories where questions that had puzzled humanity for centuries were being systematically investigated. Chemistry was emerging as a rigorous science. Physics was revealing the mathematical order underlying natural phenomena. Biology was beginning to move beyond mere description toward experimental investigation. The young Pasteur absorbed it all, attended lectures by the great chemists of the age, began the laboratory work that would occupy the rest of his life. But he also experienced Paris's other face, the crowded streets where disease spread easily, the hospitals where patients died of infections acquired after admission, the periodic epidemics of cholera and typhoid that swept through neighborhoods without explanation. The invisible enemies were all around him. Though he did not yet know to call them enemies, did not yet suspect that the air he breathed and the water he drank teemed with living things too small to see. That knowledge would come. But first, there were crystals to study, tiny geometrical forms that would provide the first glimpse into mysteries that had always been present but never perceived. We close this chapter standing with young Louis Pasteur at the threshold of his scientific career, looking toward laboratories he has not yet entered, discoveries he has not yet made, enemies he has not yet identified. The tanner's son from the Jura Mountains, the boy who drew portraits and dreamed of art, would become the man who revealed the invisible world. But that revelation lay years ahead. First, he would have to learn to see what no one else saw. First, he would have to believe in what others dismissed. First, he would have to find within himself the persistence to pursue truth wherever it led, regardless of opposition, regardless of ridicule, regardless of the comfortable assumptions that his discoveries would overturn. The village where shadows grew small, where transformation was daily miracle, where a boy learned to watch and wonder and wait. The beginning of a journey that would transform medicine, transform biology, transform humanity's understanding of the invisible world that surrounds us all. The tannery where leather emerged from raw hide, the laboratory where truth would emerge from raw observation. The same process of transformation. The same patience required. The same attention to signs that others overlooked. The village where it all began, where the smallest shadows held the largest secrets, where a child was born who would learn to see. In a Parisian laboratory, where winter light fell grey through tall windows, where the smell of chemicals hung in cold air, a young chemist held between his fingers two crystals that should have been identical, yet were not. The year was 1848. Revolutions swept through Europe, thrones toppled, barricades rose in Parisian streets. But Louis Pasteur, 25 years old and newly appointed to study under the great crystallographer Auguste Laurent, had eyes only for the tiny faceted forms that glittered in the dim light. Something was wrong with tartaric acid. Something was strange about its crystals, and that strangeness would become the first thread in a tapestry that would eventually reveal the invisible world. Tartaric acid came from wine sediment, the crusty deposits that formed inside barrels as wine aged. When dissolved and recrystallized, it formed beautiful faceted crystals that possessed a peculiar property. They rotated polarized light. Shine light through a polarizing filter, then through a solution of tartaric acid, then through another polarizing filter, and the light emerged at an angle, twisted, turned, rotated by something in the molecular structure of the acid. The phenomenon was called optical activity, and it fascinated the chemists of the age. But there was a puzzle. Sometimes tartaric acid formed crystals that looked identical but did nothing to polarized light. This inactive form, called paratartaric acid or racemic acid, was chemically identical to ordinary tartaric acid. Same elements, same proportions, same reactions. Yet one rotated light and the other did not. Why? Pasteur examined the crystals with the patience he had learned in his father's tannery, examined them with the observational skill he had developed as a young artist, and he noticed something that others had overlooked. The crystals of tartaric acid were not perfectly symmetrical. They had tiny facets on one side that were absent from the other, a subtle asymmetry, barely visible, easily dismissed as imperfection. But Pasteur did not dismiss it. He looked at the inactive paratartaric acid and found that its crystals also had these tiny facets. But some crystals had them on the left side, others on the right. The two forms were mirror images of each other, like left and right hands, identical in composition, but opposite in orientation. With tweezers and infinite patience, working under magnifying glass in the cold laboratory, Pasteur separated the crystals by hand, sorted them into two piles, those with left-facing facets, those with right-facing facets, dissolved each pile separately, tested each solution with polarized light, and discovered that the left-facing crystals rotated light to the left, while the right-facing crystals rotated light to the right. The inactive paratartaric acid was simply a mixture of the two forms. Their optical rotations canceled each other out, producing no net effect. But separated, each form was optically active. The discovery was molecular chirality, the revelation that molecules themselves could be asymmetric, could exist in mirror image forms that were chemically identical but geometrically distinct. The concept seems almost obvious today, but in 1848 it was revolutionary. It suggested that the three-dimensional arrangement of atoms mattered, that molecular structure was not just about which atoms were present, but about how they were arranged in space. It opened new fields of chemistry that would lead eventually to understanding the structure of DNA, the mechanisms of enzymes, the action of drugs on their targets. But Pasteur saw something deeper still. He noticed that living things produced only one form of tartaric acid, the right rotating form. The left rotating form appeared only when chemists synthesized the compound artificially. Life, it seemed, had a preference. Life built its molecules with a particular handedness, chose one mirror image over the other for reasons that remained mysterious. The distinction between living chemistry and dead chemistry, between the products of organisms and the products of laboratory synthesis, was written in the geometry of molecules themselves. The excitement of the discovery overwhelmed the young chemist. Legend tells that he rushed from the laboratory into the halls of the École Normale, grabbed an assistant, embraced him, and exclaimed that he had just made a great discovery. He wrote immediately to his father, "I am on the verge of mysteries, and the veil which covers them is getting thinner and thinner." The language was prophetic. Veils would continue to thin throughout his career. Mysteries would continue to reveal themselves. The crystal that turned toward light was the first thin place in the curtain that separated the known from the unknown. The scientific establishment recognized the achievement. Pasteur received the prize of the Pharmacy Society, was elected to the Legion of Honor at age 26, was appointed professor of chemistry at the University of Strasbourg. His career was launched. His reputation was established. But more important than prizes or positions was what the discovery had taught him about how to see. The tiny facets that others had overlooked, the subtle asymmetry that others had dismissed. These had held the secret. Patient observation, attention to detail, willingness to take seriously what seemed trivial, these would become his methods, would lead him from crystals to fermentation, to germs, to vaccines. In Strasbourg, Pasteur met Marie Laurent, daughter of the university rector. He courted her with characteristic intensity, wrote letters declaring his devotion, married her within a year of their meeting. She would be his partner for the rest of his life, not merely as wife and mother of his children, but as assistant in the laboratory, secretary who recorded his notes, support through the controversies and tragedies that lay ahead. Their marriage would produce 5 children, of whom 3 would die in childhood of typhoid fever. The invisible enemy that Pasteur would later learn to name, but could not save his own children from. The personal tragedy would fuel the professional determination. The crystals that turned toward light had revealed that the invisible world held secrets. The deaths of his daughters would reveal that the invisible world also held enemies. But those tragedies lay in the future. In 1849, the young professor and his bride looked toward years of peaceful research, toward laboratories where crystals would yield their mysteries, toward the slow accumulation of knowledge that characterized 19th century science. Pasteur could not know that his crystallographic work would soon be interrupted, that industrial problems would draw him away from pure chemistry toward investigations that would transform the world. Could not know that the next mystery would arrive not in geometric forms of acid crystals, but in clouded vats of fermenting beet juice, where something had gone terribly wrong. We close this chapter watching light pass through Pasteur's crystal solutions, watching the polarized beam rotate as it encounters molecules arranged with particular-handedness, watching the first evidence emerge that life and chemistry differ in ways that matter, that the living world possesses characteristics the mineral world lacks. The crystal that turned toward light had spoken, had whispered that deeper mysteries awaited, had promised that patient observation would continue to be rewarded with revelation. The veil was growing thinner. The invisible world was beginning to show itself, and the young chemist who had learned to see what others overlooked was ready to continue looking, ready to follow wherever the evidence led, ready to discover what no one yet suspected was waiting to be discovered. In the industrial cellars of Lille, where great copper vats held thousands of liters of beet juice destined to become alcohol, something had gone terribly wrong. The year was 1856. Louis Pasteur, now 33 years old and professor at the University of Lille, had been summoned by a distiller named Bigot, whose fermentations were failing inexplicably. Vat after vat was souring, producing acid instead of alcohol, turning potential profit into certain loss. The distillery faced ruin. The science of the age offered no explanation. Fermentation was understood as purely chemical process, the breakdown of sugar into alcohol through molecular transformation that required no living agent, no biological intervention. Yet the chemistry seemed to be going wrong, and no chemist could say why. Pasteur descended into the cellars with his microscope. The instrument was not new. Microscopes had existed for 2 centuries, but few chemists used them. Few thought to look at chemical processes through magnifying lenses. Chemistry was about molecules, about reactions between substances, about transformations that could be written as equations. What could a microscope reveal that equations could not? But Pasteur had learned from his crystallographic work that small details mattered, that what others overlooked might hold the key to mysteries that seemed insoluble. He would look where others had not bothered to look. He would see what others had not thought to see. The healthy fermentations, the vats producing good alcohol, contained countless tiny spherical bodies, yeasts, visible under magnification as round cells that clustered and multiplied, that seemed to be alive, that were clearly growing in the fermenting liquid. The failing fermentations, the vats producing acid instead of alcohol, contained different organisms, rod-shaped bodies, longer than the spherical yeasts, different in form, and presumably different in function. The correlation was striking. Where spherical yeasts dominated, alcohol appeared. Where rod-shaped organisms dominated, acid appeared. Fermentation was not purely chemical, it was biological. Living things performed the transformation. The conclusion seems obvious today, but in 1856 it contradicted the dominant scientific theory. The great German chemist Justus von Liebig had declared fermentation to be mechanical, a vibration of molecules that required no life, that could be understood entirely through chemistry. Yeasts were merely incidental, were present in fermentation, but not responsible for it, were byproducts rather than agents. Pasteur's observations challenged this view directly. If fermentation were purely chemical, why would different organisms produce different products? Why would the presence or absence of particular microbes determine whether sugar became alcohol or acid? Pasteur began systematic experiments, grew yeasts in controlled conditions, demonstrated that fermentation occurred only when yeasts were present and healthy, showed that different microorganisms produced different fermentation products, that the tiny creatures were performing the work, were consuming sugar, were excreting alcohol or acid as their waste products. The revelation was profound. Alcohol, humanity's ancient companion, was not created by chemistry alone. It was the excrement of living things too small to see. Every glass of wine, every mug of beer, Every dram of spirits was collaboration with the invisible, was partnership between human art and microbial labor that humans had never suspected. The implications extended beyond mere scientific interest. If fermentation was biological, it could be controlled biologically. If particular organisms produced particular products, then favoring the right organisms would produce the right results. Pasteur developed practical methods for the distillers of Lille, techniques for ensuring that proper yeasts dominated fermentations, for preventing contamination by acid-producing bacteria, for producing consistent results from inconsistent raw materials. The industrial problem was solved. The distillery was saved. But more important than saving a single business was the principle that had been established. Microscopic organisms performed chemical transformations that affected human life. The invisible world was not merely present. It was active. It was powerful. It was consequential. The discovery led naturally to questions about other transformations. If fermentation was biological, what about decay? What about the souring of milk, the rotting of meat, the spoilage that plagued food storage? Were these too the work of invisible organisms? Pasteur investigated wine that had turned sour during transport, found bacteria responsible for the unwanted fermentation, developed method of heating wine briefly to kill the contaminating organisms without damaging the wine itself, a process that would later be called pasteurization and would be applied to milk and other perishable foods, saving countless lives by preventing transmission of disease-causing microbes. But the fermentation studies raised a deeper question. Where did the yeasts and bacteria come from? They appeared in fermentation vessels, as if from nowhere. They multiplied in organic matter that had been pure. They seemed to arise spontaneously from the substances they inhabited. The ancient doctrine of spontaneous generation, the belief that life could arise from non-living matter, that maggots emerged from meat, that mice emerged from grain, that microorganisms emerged from the substances they colonized, had been challenged but not definitively refuted. Francesco Redi had shown in the 17th century that maggots did not appear in meat protected from flies, but that demonstration applied to visible creatures. What about the invisible? Could microscopic life arise spontaneously in ways that macroscopic life could not? The question was not merely scientific. It had philosophical and religious implications. If life arose spontaneously, if organization emerged from chaos without divine intervention, then the materialist philosophers were correct. Life was merely matter arranged in particular ways, requiring no creator, no soul, no spark of the divine. If life came only from life, if the continuity of living things was unbroken, then creation remained mysterious, remained possibly miraculous, remained evidence of something beyond mere chemistry. Pasteur, a man of faith, had personal stake in the outcome. But he was also a man of science who would follow evidence wherever it led. The fermentation vessels had revealed their hidden architects, the yeasts that performed alcohol fermentation, the bacteria that performed acid fermentation, the microorganisms that had worked in secret since humanity first stored food and made wine. The vessels had shown that the invisible world was populated by countless creatures pursuing their own existence, transforming their environment in the process, shaping human life without human knowledge. But the vessels had also posed a question that demanded answer. Where did these architects come from? Did they arise from nothing? Did life require life? The famous swan neck flasks were waiting to be designed. The doctrine of spontaneous generation was waiting to be tested. The invisible world had revealed itself to be alive. Now it would reveal whether that life had origins or appeared from nothing. We close this chapter in the cellars of Lille, watching fermentation proceed in vats where yeasts and bacteria perform their ancient work, watching the microscopic performers who had labored unrecognized since humanity first made wine, watching the invisible world become visible through lenses that revealed what had always been present but never seen. The fermentation vessels spoke of life in unexpected places, of transformation through biological agency, of partnership between humanity and microorganisms that humanity had never acknowledged. The hidden architects had been found, their role had been identified, their products had been traced. Now the deeper question beckoned. Where did the architects themselves originate? The vessels could not answer that question, but the flasks with curved necks would. In the quiet of his laboratory at the École Normale Supérieure in Paris, Louis Pasteur constructed instruments of elegant simplicity to answer a question that had troubled philosophers for millennia. The year was 1859, the same year Darwin published On the Origin of Species, the same year that scientific understanding of life was transforming on multiple fronts. While Darwin asked how species changed over time, Pasteur asked how life began in any particular place. Did organisms arise spontaneously from non-living matter? Or did life come only from life? Did the yeasts and bacteria that appeared in fermentation vessels emerge from nothing? Or did they descend from parents as surely as children descended from mothers and fathers? The doctrine of spontaneous generation had ancient roots. Aristotle had observed that maggots appeared in decaying meat, that frogs seemed to emerge from mud, that life proliferated wherever conditions were suitable. The appearance suggested the reality, life arising from non-life, organization emerging from chaos, the living generated spontaneously by the dead. The theory had been challenged by Francesco Redi's 17th century experiments showing that maggots did not appear in meat protected from flies, had been further challenged by Lazzaro Spallanzani's 18th century work showing that boiled broths remained sterile if properly sealed. But defenders of spontaneous generation had objections. Perhaps boiling destroyed some vital force necessary for life to emerge. Perhaps sealing prevented some essential element of air from reaching the broth. The experiments were inconclusive. The doctrine survived. The debate continued. Pasteur designed his experiments to address these objections. His famous swan neck flasks were brilliant in their simplicity. He took ordinary glass flasks, filled them with nutrient broth, heated the necks until they could be drawn out into long, thin tubes, then bent these tubes into S-shaped curves, like the necks of swans, elegant and purposeful. The flasks were boiled to sterilize the broth inside, then allowed to cool. Air could enter freely through the open tubes. No seal prevented contact with the atmosphere. No vital force was excluded. But the curved tubes trapped dust particles in their bends. The dust, carrying whatever invisible seeds might produce life in broth, settled in the curves and could not reach the sterile liquid within. The results were definitive. Broths in ordinary flasks quickly grew cloudy with microbial life. Broths in swan neck flasks remained clear for days, for weeks, for months, for years. The same broth, the same air, the same conditions, but the curved neck prevented the dust from reaching the liquid, and without the dust, no life appeared. When Pasteur tipped a flask so that broth washed through the dusty curve and back into the body of the flask, the broth quickly became cloudy with growth. The dust carried the seeds of life. Remove the dust, and nothing grew. The demonstration was dramatic, was public, was conclusive. Pasteur presented his findings to the French Academy of Sciences, challenged his opponents to explain results that their theory could not accommodate. The spontaneous generation hypothesis required that life arise from non-living matter under appropriate conditions. The conditions in the swan neck flasks were appropriate: nutrient broth, proper temperature, contact with air. Yet no life arose. The only variable was the dust, and the dust contained microorganisms that served as parents to the organisms that grew when they reached the broth. Life came from life. The continuity was unbroken. The opposition was fierce. Félix Archimède Pouchet, a prominent French naturalist, insisted that spontaneous generation occurred and published extensive experiments claiming to demonstrate it. The debate became public controversy, covered in newspapers, discussed in cafes, implicating not just scientific questions, but philosophical and religious positions. Materialists wanted spontaneous generation to be true. It supported their view that life required no creator, that matter could organize itself. Religious believers wanted it to be false. It preserved the mystery of life's origin, left room for divine creation. Pasteur walked a careful line. insisted that his experiments proved nothing about ultimate origins, showed only that life came from life within observable conditions, left philosophical and theological conclusions to philosophers and theologians. But the experiments had practical implications that extended beyond philosophy. If microorganisms came from particles floating in the air rather than arising spontaneously, then preventing contamination was possible. Sterile broth remained sterile if protected from dust. Wine remained uncontaminated if sealed properly. Surgical wounds would not become infected if kept clean. The germ theory of disease, the idea that specific microorganisms caused specific diseases, became conceivable once spontaneous generation was disproved. If microbes arose spontaneously, they were merely symptoms of disease, products of decay, consequences rather than causes. If microbes came only from other microbes, they could be causes themselves, could be transmitted from person to person, could be prevented through cleanliness and isolation. Pasteur took his experiments to the mountains. If the air carried particles that produced life in broth, those particles should be more abundant in densely populated places and less abundant in remote locations. He traveled to the Alps with sealed flasks of sterilized broth, opened them briefly at various altitudes, sealed them again, brought them back to Paris. The flasks opened in the valley often became cloudy with growth. The flasks opened at high altitude often remained clear. The air varied. The particles varied. The distribution of life's invisible seeds varied with location, with human presence, with the density of living things that produced the particles. The swan neck flasks became icons of scientific method, elegant experiments that isolated variables, controlled conditions, produced unambiguous results. Some of Pasteur's original flasks survive, are displayed at the Pasteur Institute, their broths still clear after more than 150 years. The longevity testifies to the success of the design, to the effectiveness of the dust trap, to the proof that sterile conditions remain sterile when contamination is prevented. The flasks are relics now, objects of scientific pilgrimage, instruments that settled a debate that had persisted for millennia. But the significance extended beyond settling an academic dispute. The death of spontaneous generation cleared the way for understanding disease as the result of specific causes that could be identified and combated. If life came only from life, then the organisms infecting wounds and spoiling food and killing children had sources that could be traced, routes of transmission that could be interrupted, methods of prevention that could be developed. The invisible enemies had not only been revealed, their mode of propagation had been established. They reproduced, they spread, they could be stopped. We close this chapter, contemplating the swan neck flasks in their serene clarity, their broths still sterile after century and a half, their silence speaking louder than any proliferation could. The flask with the curved neck had answered the ancient question. life comes from life. The continuity is unbroken. The seeds of the invisible world are carried on dust and air and water, are deposited wherever conditions favor growth, are absent only where physical barriers prevent their arrival. The doctrine that had endured since Aristotle died in glass vessels, shaped like questions. The way was clear for understanding disease. The way was clear for developing vaccines. The way was clear for the revolution in medicine that would save millions of lives. All because a chemist shaped glass tubes into curves that trapped dust. All because someone thought to ask where the invisible creatures came from. All because elegant experiment answered eternal question. Across the mulberry groves of southern France, a catastrophe was unfolding in silence. The year was 1865. The silkworms were dying. Those delicate larvae whose silk glands had clothed emperors for millennia, whose cocoons had made fortunes for generations of farmers, whose cultivation was art passed from parent to child across centuries. The disease appeared without warning. Worms that had seemed healthy blackened with pepper-like spots, their bodies withering, their silk glands failing, their cocoons incomplete or never begun. The plague called pébrine had devastated France's silk industry. Production had collapsed to a fraction of former levels. Farmers faced ruin. An industry that had flourished since the Romans faced extinction. The authorities were desperate. They summoned Louis Pasteur, whose work on fermentation and spontaneous generation had established his reputation as scientist who could solve practical problems. But Pasteur knew nothing of silkworms, had never raised them, had never studied them, had never given them more than passing thought. When he accepted the commission, prompted partly by a former teacher who was ill and could not undertake it himself, he reportedly asked whether silkworms even had cocoons. The ignorance was genuine. The confidence that he could overcome it was characteristic. Pasteur moved his family to Alais in the south of France, established himself among the mulberry groves and the dying worms, began the painstaking work of observation that had served him in every previous investigation. What he found was more complex than he had anticipated. The disease called pébrine was not one disease, but two. Separate conditions with different causes, different symptoms, different modes of transmission. The confusion had hindered all previous attempts at understanding. The farmers saw worms dying and called it pébrine without distinguishing the distinct afflictions that were claiming their creatures. The first disease, the true pébrine, was caused by a microscopic parasite, a protozoan that Pasteur could see within the cells of infected worms, that multiplied as the disease progressed, that was transmitted from mother moth to eggs, ensuring that the next generation was infected before it was born. The pepper-like spots that gave the disease its name were concentrations of the parasite visible through the worm's translucent skin. The disease was hereditary in a sense, not genetic inheritance, but the parasite passing from generation to generation within the eggs themselves. The second disease, which Pasteur named flacherie, was caused by bacteria in the digestive tract. Worms with flacherie grew soft and flaccid, their intestines fermenting with microbial overgrowth, their bodies literally rotting from within while still alive. This disease was not hereditary, but was spread through contaminated food, through droppings that infected mulberry leaves, through the general environment of the silk-raising houses. The two diseases often appeared together, their symptoms overlapping, their combined assault devastating the silk industry that could survive neither alone. The research was exhausting, was lonely, was shadowed by personal tragedy. During the years Pasteur spent studying silkworms, 2 of his daughters died of typhoid fever. His own health began to fail. The first strokes that would eventually paralyze half his body struck during this period, leaving him weakened, aged before his time. The microscope that revealed parasites in silkworms could not reveal the bacteria killing his children. The knowledge that was slowly emerging about disease causation could not yet be applied to save the lives that mattered most to him. But the work continued. Pasteur developed practical methods for the silk farmers, techniques for identifying infected moths by examining them microscopically after they laid their eggs, for selecting only healthy stock for breeding, for eliminating the hereditary transmission of pébrine by starting each generation from certified disease-free parents. The methods worked. The industry recovered. French silk production rose again as farmers adopted Pasteur's protocols, as microscopic diagnosis became standard practice, as the invisible enemy was identified and controlled. The silkworm research taught lessons that extended far beyond sericulture. It demonstrated that infectious diseases could be diagnosed through microscopic examination, that healthy and sick could be distinguished by the presence or absence of specific organisms, that epidemics could be controlled through understanding rather than superstition. The techniques developed for silkworms would later be applied to human diseases, the identification of pathogenic organisms, the tracing of routes of transmission, the development of preventive measures based on scientific understanding. The dying worms had been teachers, their plague a lesson in methodology that would save human lives. But the silkworm years had cost Pasteur dearly. His health never fully recovered. The strokes left lasting damage, weakened him, aged him prematurely. The loss of his daughters haunted him. The knowledge that typhoid fever had claimed them, that bacteria had killed the children of the man who was learning to identify bacteria, that his own family had suffered from the very enemies he was teaching the world to fight. The personal tragedy fueled the professional determination. Pasteur would spend the rest of his career fighting invisible enemies, would develop vaccines and techniques that would save millions of lives, would perhaps be driven by the knowledge that he had not been able to save the lives that mattered most. We close this chapter in the mulberry groves of southern France, watching the silkworms spin their cocoons in the lamplight, watching the industry that had nearly died recover through application of scientific understanding, watching the man who had identified the invisible parasites weigh his triumph against his losses. The threads of ruin had been rewoven into threads of silk. The plague that had devastated the industry had become the teacher that revealed the nature of infectious disease. But the lessons had come at terrible cost. Daughters buried, health broken, the man himself transformed by what he had witnessed and what he had lost. The silkworm plague, the threads of ruin, the knowledge purchased through suffering, forever shaping what would come next. In hospital wards across Europe, a horror was unfolding that no one yet understood. The year was 1867. Patients who survived surgery frequently died anyway, not from the knife wounds themselves, which had been cleanly made and properly sutured, but from infections that set in afterward, from fevers that rose without explanation, from gangrene that blackened flesh that should have healed, from sepsis that poisoned blood that should have remained pure. Surgeons operated in street clothes, their frock coats stiff with the dried blood of previous patients. The stiffness was badge of honor, evidence of experience, mark of the busy practitioner. Hands were washed casually, if at all. Instruments were reused with only cursory cleaning. The wards smelled of decay because decay was constant, was expected, was simply the price of surgical intervention. In Edinburgh, a surgeon named Joseph Lister had been reading Pasteur's work on fermentation and spontaneous generation, had been thinking about what it might mean for wounds that festered. If invisible organisms caused wine to sour, could they also cause wounds to rot? If life came only from life, then the organisms infecting surgical wounds must come from somewhere, from the air, from the instruments, from the hands that touch the patients. Could preventing their arrival prevent the infection? Lister began treating surgical wounds with carbolic acid, a substance derived from coal tar that was known to eliminate the smell of sewage, presumably by killing whatever caused the smell. He sprayed carbolic acid in the operating theater, soaked dressings in it, applied it to wounds and instruments. The results were dramatic. Infection rates plummeted. Patients who would have died of gangrene survived. Wounds that would have festered healed cleanly. The invisible enemies could be killed with chemicals. The infections could be prevented through antiseptic practice. The connection between Lister's antisepsis and Pasteur's microbiology was explicit. Lister wrote to Pasteur acknowledging the debt, thanking him for the scientific foundation that had made antiseptic surgery conceivable. Pasteur was moved by the tribute, recognized that his laboratory work was finding practical application in saving lives, understood that the chain from crystals to fermentation to spontaneous generation to disease prevention was proving its value in hospital wards where patients lived who would otherwise have died. But the medical establishment resisted. Surgeons who had practiced for decades without antiseptic precautions dismissed the new ideas as nonsense. The germ theory of disease seemed absurd. How could tiny creatures invisible to the naked eye kill grown men? The miasma theory, which blamed disease on bad air arising from decay, had the weight of tradition behind it. The idea that doctors themselves might be spreading disease through unwashed hands was offensive to professional pride. Ignaz Semmelweis, a Hungarian physician who had demonstrated in the 1840s that hand-washing could prevent childbed fever, had been ridiculed, driven from his position, eventually died in a mental asylum. The resistance to germ theory was fierce, was personal, was sometimes violent. Yet the evidence accumulated. Robert Koch in Germany developed techniques for isolating and identifying specific bacteria, proved that particular organisms caused particular diseases, discovered the bacterium responsible for anthrax in 1876, the bacterium responsible for tuberculosis in 1882, the bacterium responsible for cholera in 1883. Each discovery strengthened germ theory, weakened alternative explanations, built the case for understanding disease as consequence of infection by specific microbes rather than as result of miasma or humoral imbalance or moral failing. Pasteur himself entered the medical arena increasingly as his career progressed. The silkworm work had been agricultural, had saved an industry rather than human lives, but the principles were transferable. If microscopic parasites could cause disease in worms, microscopic organisms could cause disease in humans. If selective breeding could eliminate inherited infection in silkworms, perhaps vaccination could eliminate susceptibility to infection in people. The invisible war had been declared, humanity against microbes, a war that had been fought unconsciously since humans first fell ill, but was now being fought with knowledge, with understanding, with weapons developed through scientific investigation. The war would not be won quickly. Even today, infectious diseases remain major killers. New pathogens emerge to challenge medical defenses. Old enemies develop resistance to drugs that once controlled them. But the declaration of war, the recognition that disease had causes that could be identified, that identification enabled prevention and treatment, that the invisible world was enemy territory to be mapped and conquered, this transformation in thinking was fundamental. Before Pasteur and Koch and Lister, disease was fate. After them, disease was challenge. The difference has saved countless millions of lives. We close this chapter contemplating the invisible war in its early years, when the enemy had just been identified, when the first victories were being won, when surgeons like Lister were proving that antiseptic practice could prevent infections that had seemed inevitable. The war continues still. New enemies emerge, viruses, antibiotic-resistant bacteria, pathogens not yet named. But the fundamental understanding remains. Disease is caused by specific agents that can be identified and fought. The invisible war declared in the 19th century has no foreseeable end. But every day, in laboratories and hospitals around the world, the battle continues that Pasteur's work made possible. The enemies are real. The war is real. The victories, though never permanent, are real as well. In a field near the village of Pouilly-le-Fort, 50 sheep stood divided into 2 groups on a spring morning in 1881. 25 vaccinated with Pasteur's experimental preparation, 25 left unprotected. The demonstration that would unfold over the following days would prove principles that would transform medicine, would save millions of lives, would establish vaccination as humanity's most powerful weapon against infectious disease. But on that May morning, nothing was certain. Pasteur had staked his reputation on an experiment whose outcome he could not fully predict. The vaccinated sheep might die alongside the unvaccinated, revealing his methods as worthless. Or they might live, proving that weakened pathogens could teach the body to resist their deadly relatives. Anthrax was terror. The disease struck livestock without warning, killed swiftly and horribly, left carcasses blackened in pastures, could devastate herds within days. The bacterium responsible, Bacillus anthracis, had been identified by Robert Koch in 1876, one of the first demonstrations that specific microbes cause specific diseases. But identification was not prevention. Knowing the enemy did not defeat it. Farmers watched helplessly as their animals died, burned the carcasses to prevent spread, knew that the disease could return at any time, could strike humans as well as animals, could kill the farmers themselves if they handled infected material. Pasteur had been working on anthrax for several years, seeking to understand how the disease spread, how the bacterium survived in soil for decades, how it might be prevented. His approach was inspired by Edward Jenner's smallpox vaccination, the 18th century discovery that infection with cowpox, a mild disease, protected against smallpox, a deadly one. Could similar protection be developed for anthrax? Could the deadly bacterium be weakened somehow, made harmless but still capable of teaching the immune system to recognize and destroy its virulent relatives? The method Pasteur developed, or that his assistants developed, for the credit remains disputed, involved exposing anthrax bacteria to oxygen at elevated temperatures. The exposure weakened the organisms, made them less lethal, produced strains that could infect animals without killing them. Animals that recovered from infection with these weakened strains were protected against subsequent infection with virulent anthrax. The immunity could be created artificially. Vaccination against anthrax was possible. The public demonstration at Pouilly-le-Fort was Pasteur's answer to skeptics who doubted his claims. The challenge had been issued by a veterinarian named Hippolyte Rossignol, who had published articles mocking germ theory and vaccination. Pasteur accepted the challenge boldly, perhaps rashly, for the vaccine had not been fully tested, the outcomes were not guaranteed, public failure would have been devastating to his reputation and his cause. On May 5th, 25 sheep were vaccinated with a weakened anthrax preparation. On May 17th, they received a second vaccination with a slightly stronger preparation. 25 control sheep received no vaccination. On May 31st, all 50 sheep received injections of virulent anthrax, doses that would normally be fatal. The assembled observers, farmers, veterinarians, journalists, scientific skeptics, watched and waited. 2 days later, on June 2nd, the results were unambiguous. The 25 vaccinated sheep stood healthy in the morning light, grazing peacefully, showing no signs of illness. Of the 25 unvaccinated sheep, 22 were dead, 2 were dying, and 1 appeared mortally ill. The demonstration was complete, was public, was undeniable. The press reported the results across Europe. Pasteur was acclaimed as hero, as savior of agriculture, as scientist whose laboratory work had produced practical miracle. But the triumph contained complications that would only emerge later. Evidence suggests that the actual vaccine used at Pouilly-le-Fort was not the oxygen-attenuated preparation Pasteur had publicly described, but a chemically treated vaccine developed by his assistant, Charles Chamberlain, using methods pioneered by another researcher, Henri Toussaint. The discrepancy may have been intentional concealment of methods Pasteur was not ready to publish, or may have been substitution made because the oxygen-attenuated vaccine was not reliable enough for public demonstration. The full truth remains unclear. Pasteur's laboratory notebooks, examined by historians decades later, reveal contradictions with his public statements that have never been fully resolved. Does the controversy diminish the achievement? The questions about methods are legitimate. The concerns about scientific honesty are important. But the sheep did not lie. The vaccinated lived. The unvaccinated died. The principle of vaccination was demonstrated conclusively, whatever the specific technique employed. The anthrax fields of Pouilly-le-Fort witnessed something unprecedented: deliberate, controlled protection against a deadly disease through administration of weakened pathogens. The principle would be applied to rabies, to cholera, to typhoid, to diphtheria, to diseases that had killed millions for millennia. The methods mattered. The ethics mattered. But the outcome also mattered. And the outcome was salvation. We close this chapter in the anthrax fields at sunrise, watching vaccinated sheep graze peacefully, while the unvaccinated lie dead around them, watching the moment when vaccination proved its power, when the invisible enemies were turned against themselves, when the weakness of pathogens became strength of protection. The fields bore witness to triumph and to controversy, to principle proven and to methods disputed, science that saved lives even when its practitioners failed to meet every ethical standard. The anthrax fields of Pouilly-le-Fort, forever testimony, forever complicated, forever the moment when vaccination entered its modern age. On July 6, 1885, a 9-year-old boy named Joseph Meister arrived at Louis Pasteur's laboratory in Paris, his small body bearing 14 wounds from a rabid dog. The attack had occurred 2 days earlier in Alsace, the boy walking to school when a mad dog knocked him down, bit him repeatedly on hands and legs and thighs before a bricklayer drove the animal away. The dog was killed, its stomach open to reveal straw and hay, evidence that it had been rabid, for mad dogs often swallowed strange objects. The boy's mother had heard of Pasteur's work on rabies, had brought her son by train to Paris, had arrived desperate at the laboratory of the only man who might save him. Rabies was death sentence. Once symptoms appeared, the fever, the hydrophobia, the madness, the paralysis, no one recovered. The virus traveled slowly along nerve fibers from the bite wound to the brain, taking weeks or months depending on the wound's location. During this latent period, the victim seemed healthy, went about daily life, knowing that each day might be the last before symptoms appeared and death became inevitable. The terror was as much psychological as physical, the knowledge that something was growing within, that death approached invisibly, that nothing could be done. Pasteur had been working on rabies for years. Unlike anthrax, which could be grown in laboratory flasks, the rabies agent could not be cultivated outside living tissue. Pasteur did not know he was dealing with a virus, organisms too small to see with light microscopes, requiring electron microscopes invented decades later. But he knew that something in the saliva of rabid animals transmitted the disease, that this something affected the nervous system, that it could be passed from animal to animal through injection. His method for creating a vaccine was ingenious. He took spinal cords from rabbits that had died of rabies, suspended them in dry air for varying periods. Fresh cords contained virulent virus. Cords dried for days or weeks contained weakened virus. By administering a series of injections starting with the weakest preparation and progressing to stronger ones, he hoped to teach the immune system to resist rabies before the virus naturally present in the bite victim could reach the brain. The method had worked in dogs. Animals vaccinated with the dried cord preparations survived bites from rabid dogs that killed unvaccinated controls. But dogs were not humans. Pasteur was not a physician. He had no legal authority to treat patients. The vaccine might work differently in human tissue. It might cause the disease rather than prevent it. It might do nothing while the virus progressed inexorably toward the brain. Treating Joseph Meister was gamble that could end Pasteur's career, could end his freedom if the boy died and he was charged with causing the death, could end everything he had built. But refusing to treat was also choice. Joseph would almost certainly die of rabies. The wounds were severe. The dog was clearly rabid. The virus was almost certainly already traveling toward the boy's brain. Doing nothing meant certain death. Attempting treatment meant possible salvation with risk of acceleration. Pasteur consulted with physicians, weighed the options, decided. Over 11 days, Joseph received 13 injections of rabies-infected spinal cord preparations, starting with material dried for 14 days and progressing to fresher, more virulent material. The final injection contained preparation dried for only 1 day, nearly as virulent as fresh material. If Joseph's immune system had not developed resistance through the earlier injections, this final dose might kill him. Pasteur barely slept during the treatment, checked on Joseph constantly, worried that he had made terrible mistake, that the boy would die and it would be Pasteur's fault, that the treatment was more dangerous than the disease. But days passed. Weeks passed. 3 months passed, longer than any incubation period ever recorded. Joseph Meister lived. He never developed rabies. The vaccine had worked. The news spread across the world. Newspapers acclaimed Pasteur as miracle worker, as savior, as scientist who had conquered the most terrifying disease known to humanity. Victims of rabid animal bites traveled from across Europe, from Russia, from America, to receive the treatment. A Russian peasant named Ivan arrived months after being bitten by a rabid wolf. His wounds had healed, but he knew the virus was within him, traveling toward his brain. Pasteur treated him and 17 other Russians who had been attacked by the same wolf. 16 survived. The deaths of the 3 who did not survive haunted Pasteur. Were they too late to save, or did the treatment fail? Joseph Meister survived to adulthood, became gatekeeper at the Pasteur Institute, spent his life at the institution that bore the name of the man who had saved him. In 1940, when German soldiers occupying Paris demanded he open Pasteur's crypt, Meister refused, then committed suicide rather than comply. The boy from Alsace died protecting the tomb of the man who had saved his life. The connection between scientist and patient, forged in desperate experiment, endured across half a century. We close this chapter watching Joseph Meister receive his final injection, watching Pasteur wait anxiously for symptoms that never came, watching the moment when rabies ceased to be death sentence, when the most terrifying disease of the age yielded to scientific understanding. The boy who came from Alsace became the first human saved by vaccination against a disease already contracted. The gamble succeeded. The principle was proven. The door was opened for vaccines against diseases not yet known, against enemies not yet identified, against the invisible world that would continue to threaten humanity, but would never again threaten without response. Joseph Meister, the first of millions, the boy who lived. On the Rue du Tau in Paris, a building rose that would house humanity's ongoing war against the invisible. The year was 1888. Public subscription had raised the funds, francs donated by citizens grateful for the rabies treatment, for the anthrax vaccine, for the fermentation studies that had saved the wine and beer industries, for the silkworm work that had restored sericulture. The donations came from princes and from peasants, from scientists and from farmers, from across France and from across the world. The Pasteur Institute would be temple and fortress, laboratory and hospital, training center and research station, all dedicated to the continuing battle against microscopic enemies. Pasteur himself, weakened by strokes that had paralyzed half his body, presided over the inauguration. He walked with difficulty, spoke with difficulty, his left side unresponsive, his face asymmetric. He was 65 years old, looked older, had driven himself through decades of labor that had broken his health while building his legacy. The ceremony must have been bittersweet, triumph tempered by mortality, achievement shadowed by decline, the knowledge that others would continue work he could no longer perform himself. The Institute's founding represented new model for scientific research. Dedicated institution where scientists could pursue investigations free from teaching obligations, where international staff could collaborate across national boundaries, where practical applications were developed alongside fundamental discoveries. The model would be imitated worldwide. Research institutes bearing names of other scientists would follow. Koch Institute in Berlin, Rockefeller Institute in New York, all inspired by the precedent Pasteur established. The researchers who gathered at the Institute continued the work their master had begun. Émile Roux and Alexandre Yersin discovered diphtheria toxin, developed anti-toxin treatment that saved children from the strangling disease. Albert Calmette and Camille Guérin developed BCG vaccine against tuberculosis. Alexandre Yersin traveled to Hong Kong during bubonic plague outbreak, identified the bacterium responsible. Charles Nicolle at the affiliated institute in Tunis identified the louse as vector for typhus. The discoveries flowed from Pasteur's foundations, each building on principles he had established, each extending understanding of the invisible world. But the Institute was more than laboratory, was also clinic where victims of rabid animal bites received the treatment Pasteur had pioneered. Patients arrived daily from across France, from across Europe, from across the world. The treatment room became pilgrimage site where the desperate came seeking salvation, where injections were administered by physicians trained in Pasteur's methods, where the boundary between science and hope was crossed daily. Not all patients survived. Some arrived too late, their infections too advanced. But most did, and the Institute's statistics testified to vaccination's effectiveness. The Institute also trained the next generations of microbe hunters. Young scientists arrived to learn techniques, to work alongside the masters, to absorb the culture of careful observation and controlled experimentation that Pasteur had exemplified. They dispersed afterward across the world, founding laboratories and institutes of their own, spreading methods and principles, creating global network of researchers pursuing the invisible enemies that threatened humanity. The Pasteurians, they were called, and they carried their master's legacy to every continent. The building itself embodied the transition from individual achievement to institutional continuity. Science had been practiced by individuals, Newton alone in his study, Darwin on his estate, even Pasteur working with a handful of assistants. The institute represented collectivized research, coordinated effort, sustained investigation across generations. The problems were too large for individuals. The enemies were too numerous for single careers. The institution would continue when individuals retired or died, would maintain continuity of effort, would accumulate understanding across decades and centuries. Pasteur spent his final years at the institute, increasingly frail, increasingly dependent on his wife Marie, who had supported him throughout his career. He received visitors, scientists seeking guidance, politicians paying respects, former patients expressing gratitude. Joseph Meister, the boy from Alsace, worked at the institute, daily reminder of what had been achieved, living testimony to vaccines' power. Pasteur watched younger scientists pursue paths he had opened, knowing his work would continue beyond his failing body, trusting that the institute would preserve and extend his legacy. We close this chapter contemplating the building on Rue du Tau, its laboratories humming with work that continues today, its archives preserving records of discoveries that changed the world, its culture embodying principles Pasteur established. The institute that bears his name became living legacy. Stone and glass housing ongoing battle, generation after generation trained in its methods, discovery after discovery flowing from its laboratories. The building is modest from outside, gives no hint of the war being waged within, of the enemies being studied and sometimes defeated, of the vaccines being developed and tested and deployed. But inside, the invisible war continues. The institute stands. The work goes on. The legacy lives. On October 19th, 1868, in the midst of his silkworm research, Louis Pasteur felt the world tilt. He was sitting at dinner when the attack began. Numbness spreading through his left side, his face going slack, his speech thickening. A stroke had seized him, was paralyzing half his body, was threatening to end his career at 45 years old, at the height of his powers, in the middle of his most important work. The physicians gathered, examined, pronounced the outlook grave. Complete recovery seemed unlikely. Continued scientific work seemed impossible. But Pasteur refused to die, refused to surrender. Within weeks, he was dictating notes to his wife, continuing his silkworm research through her hands when his own would not obey. Within months, he was back in the laboratory, had learned to work with half a body while his mind remained whole. The paralysis would be permanent. His left arm would never regain function. His left leg would drag. His face would remain asymmetric. But his will would not be paralyzed. His determination would not stroke. The strokes would recur throughout his remaining years. Each attack stole something: more function, more mobility, more capacity for the physical work that laboratory science required. But each attack also revealed something: the depth of his determination, the strength of his will, the refusal to surrender to mortality while work remained undone. He continued through the anthrax vaccine development, through the rabies treatment that saved Joseph Meister, through the founding of the institute that would bear his name. Each achievement came from a man whose body was failing while his mind remained active. The personal cost of scientific devotion extended beyond physical health. During the silkworm years, 2 of Pasteur's daughters died of typhoid fever, the disease caused by bacteria, by invisible enemies that Pasteur was learning to identify but could not yet defeat. Jeanne died in 1859 at age 9. Cécile died in 1866 at age 12. The father who was teaching the world to understand infectious disease could not save his own children from infection. The irony was cruel, was perhaps motivation, was certainly shadow that haunted his subsequent work. His wife Marie bore the burden of nursing and supporting him through decades of declining health. She had married a young professor, had become partner to a genius, had watched her husband's body fail while his achievements multiplied. She transcribed his notes when he could not write, managed his correspondence when he could not travel, protected him from demands that would have exhausted his limited strength. Their partnership was essential to his productivity. Without her, his final decades of achievement would have been impossible. The parallel between Pasteur's personal condition and his scientific work is striking. He spent his career revealing the invisible, organisms too small to see, causes hidden within effects, enemies that operated beneath perception's threshold. His own condition made him partly invisible to himself, half his body foreign territory, half his sensory world extinguished, half his physical capacity stolen. The man who taught the world to see the invisible lived within invisibility, experienced daily the limitations of perception, knew in his own flesh that what cannot be seen can still be consequential. The strokes did not diminish his ambition. If anything, they increased his urgency, the knowledge that time was limited, that more attacks might come, that work needed to be completed before the body failed entirely. The rabies vaccine was developed in his 60s, while he was already partially paralyzed. The Pasteur Institute was founded when he was too weak to participate fully in its operations. The determination that had characterized his earlier career intensified as his physical capacity diminished. His final years were spent at the Institute, receiving visitors, watching younger scientists continue his work, declining gradually toward death. Marie remained beside him, reading to him, caring for him, sharing the quiet of his final months. He died on September 28th, 1895, aged 72, his hand reportedly resting in his wife's, his work completed, his legacy established. We close this chapter contemplating the man who fought invisible enemies while his own body betrayed him, who continued working while half the world was stolen from his perception, who refused to surrender to mortality until mortality finally claimed him. The stroke that stole half the world could not steal his will, could not steal his determination, could not steal the contributions he would continue to make through decades of declining health. The paralysis was limitation, was obstacle, was daily reminder of mortality, but it was not defeat. The work continued. The discoveries accumulated. The man who could not control his own body taught the world to control invisible enemies that had plagued humanity for millennia. In laboratories across Europe and beyond, a new priesthood emerged in the decades following Pasteur's revelations. Scientists who spent their lives bent over microscopes, who pursued creatures too small for unaided human sight, who mapped the invisible kingdom that had been revealed. Robert Koch in Germany developed techniques for growing bacteria in pure culture on solid media, for staining organisms to make them visible under microscopes, for fulfilling postulates that established causal connections between specific microbes and specific diseases. Joseph Lister in Scotland transformed surgery through antiseptic practice, proving that cleanliness could prevent infections that had seemed inevitable. The discipline of microbiology spread from Pasteur's work like ripples from a stone dropped in still water. Koch's contributions rivaled Pasteur's in importance. His 1876 identification of the anthrax bacillus was the first rigorous demonstration that a specific bacterium caused a specific disease. His 1882 discovery of the tuberculosis bacillus targeted humanity's greatest killer, the disease that claimed more lives than any other, that had been called consumption and the White Plague, that represented perhaps 1/4 of all deaths in 19th century Europe. His 1883 identification of the cholera vibrio during an epidemic in Egypt demonstrated the power of the new microbiology applied to epidemic disease. Koch's postulates, the criteria for establishing that a particular microbe causes a particular disease, became foundation for medical microbiology, framework that guided research for generations. The rivalry between Pasteur and Koch was intense, was personal, was sometimes bitter. The Franco-Prussian War of 1870-71 had left France humiliated, its territory diminished, its national pride wounded. Pasteur, a fierce patriot, resented German scientific achievements, saw competition with Koch as continuation of national conflict by other means. Koch, for his part, criticized Pasteur's methods, questioned his anthrax vaccine, published attacks that were sometimes petty and sometimes substantive. The scientific competition was genuine. Both men made important discoveries. Both deserved credit. But the personal animosity exceeded what scientific disagreement warranted. The cathedral that Pasteur's discoveries built was not a building, but a discipline, a way of seeing, a methodology that would identify the causes of diseases that had plagued humanity for millennia. The achievements of the late 19th and early 20th centuries were remarkable. Diphtheria antitoxin, developed at the Pasteur Institute, provided treatment for the strangling disease that had killed countless children. Plague bacillus was identified during outbreaks in Hong Kong and Bombay. Typhus vector was identified as the body louse. Yellow fever was shown to be transmitted by mosquitoes. Disease after disease yielded its secrets to the new methods. But the cathedral also had limitations that would become apparent only gradually. Germ theory encouraged focus on external enemies, bacteria and other microbes that invaded from outside, that could be identified and combated. This focus was appropriate for infectious diseases, was spectacularly successful against cholera and typhoid and diphtheria. But other diseases had other causes: genetic disorders, nutritional deficiencies, environmental toxins, immunological dysfunctions, and the germ theory framework sometimes hindered understanding of these conditions. The success against some diseases created hubris that hampered understanding of others. The technology evolved alongside the theory. Microscopes became more powerful, revealing details invisible to earlier instruments. Staining techniques allowed visualization of structures within cells. Culture methods enabled growth of microorganisms in controlled conditions. Each advance opened new questions, revealed new complexity, showed that the invisible world was far more intricate than early discoveries had suggested. The bacteria that seemed simple under early microscopes proved to have complex structures, diverse metabolisms, sophisticated behaviors. The invisible kingdom was vast beyond imagining. The implications extended beyond medicine. Food preservation was transformed by understanding of microbial spoilage. Pasteurization made milk safe to drink. Canning methods were refined to ensure sterility. Refrigeration slowed microbial growth. Agriculture benefited from understanding of nitrogen-fixing bacteria, of soil microorganisms, of plant diseases caused by microbial pathogens. Industry employed microorganisms in fermentation processes, in production of chemicals and pharmaceuticals, in applications their discoverers could not have imagined. The cathedral of microscopes rose over Pasteur's foundations, its worship precise, its sacraments sterile, its prayers whispered in Latin names for tiny enemies. The invisible world had been mapped, partially, imperfectly, with vast territories still unexplored. But the war had been organized. The enemies had been identified. The methods for fighting them had been established. and humanity began winning battles it had never before been able to fight. We close this chapter contemplating the cathedral in its full complexity, the achievements and the limitations, the triumphs and the failures, the diseases conquered and the diseases that still resist. The cathedral continues to grow, its spires reaching toward understanding we have not yet achieved, its foundations extending into territory we have not yet mapped. The microscopes have become electron microscopes, then scanning probe microscopes, then instruments that can visualize individual atoms. The invisible world keeps revealing deeper layers, smaller structures, more complex interactions. The cathedral is never finished. The worship never ends. In a Byzantine chapel within the Pasteur Institute, Louis Pasteur lies entombed. He died on September 28, 1895, surrounded by family, weakened by his final stroke, aged 72. The funeral was state occasion, France honoring its scientific hero with pomp befitting royalty. The body lay in state at Notre Dame before transfer to the institute he had founded, where it rests still in a crypt decorated with mosaics depicting his discoveries. The crypt is ornate, is beautiful, is perhaps too triumphalist for a man who valued empirical truth over ceremonial display. The mosaics show his achievements rendered in colored stone, the crystallographic work depicted in geometric patterns, the fermentation studies represented by grapes and laboratory vessels, the silkworm research shown through mulberry leaves and moths, the rabies triumph portrayed through the child Joseph Meister receiving his injections. The imagery transforms science into iconography, research into sacred narrative. The man who demanded evidence has become object of something resembling faith. Visitors descend into the quiet space, stand before the tomb of the man who revealed the invisible, who proved that life comes only from life, who demonstrated that disease has causes that can be identified and fought. They are pilgrims of a sort, scientists paying homage to a founder, students seeking inspiration, tourists curious about the famous name. The crypt offers no sermons, demands no beliefs, requires no rituals. It simply holds what remains of a man who changed the world. But the tomb also holds contradictions. The historical Pasteur was more complex than the legend, a fierce competitor who sometimes claimed credit he did not fully deserve, a nationalist whose patriotism could become chauvinism, a scientist whose methods were not always as pure as his public pronouncements suggested. The controversies about the anthrax vaccine, about the precise techniques used for rabies treatment, about credit shared or stolen with assistance. These complications are not depicted in the mosaics, are not spoken of in the commemorations, are perhaps not appropriate for the crypt of a hero. And yet the complications do not diminish the achievements. Whatever the precise methods, the vaccinations worked. Whatever the contested credit, The discoveries were made. Whatever the personal failings, the lives were saved. Millions of lives, continuing to be saved by vaccines and antiseptic practices and understanding that flowed from Pasteur's foundations. The man in the tomb was human, was flawed, was not the saint the mosaics might suggest, but the contribution was real. The legacy endures. The work continues above the crypt, in laboratories where scientists still pursue the invisible enemies Pasteur first taught humanity to see. The Institute has evolved, has expanded, has adapted to challenges its founder could not have imagined. Viruses were discovered, organisms too small for light microscopes, requiring electron microscopes developed decades after Pasteur's death. Antibiotics were developed, chemicals that killed bacteria within the body, transforming treatment of infections. Molecular biology emerged, understanding of genes and proteins and cellular mechanisms that explained how microorganisms cause disease. Each advance built on Pasteur's foundations while transcending them. The battles continue. HIV emerged in the 1980s, created pandemic that killed millions before treatments were developed. Antibiotic-resistant bacteria evolve, threaten to undo the progress antibiotics achieved. New pathogens appear: SARS, MERS, Ebola, influenza strains with pandemic potential. The invisible enemies adapt, evolve, find new ways to threaten humanity. The war Pasteur declared is not won, may never be won. The enemies are too numerous, too adaptable, too creative in their evolution. But the war can be fought, can be waged with knowledge rather than ignorance, can be conducted with vaccines and antisepsis and understanding. Joseph Meister, the boy from Alsace, served as gatekeeper of the Institute for decades. He showed visitors the laboratory where he had received his injections, told the story of his salvation to those who asked. In 1940, when German soldiers occupied Paris and demanded access to Pasteur's crypt, Meister refused to open the gates. He was not protecting mere bones, was protecting symbol of French scientific achievement, was protecting memory of the man who had saved his life. Rather than comply with the German demands, Meister committed suicide. The boy Pasteur saved died protecting Pasteur's tomb. The connection forged in desperate experiment endured across half a century, ended only in tragedy when another war, not against microbes, but against nations, claimed another victim. We close this narrative, standing in the crypt, feeling the weight of what lies there. Not just bones, but legacy. Not just remains, but reminder. The man who revealed the invisible is invisible now, his body hidden beneath stone and mosaic. The man who taught us to see what we could not see can no longer see anything. the man who conquered death through understanding, finally died as all men die. But the work continues. The Institute stands. The vaccines save lives. The understanding accumulates. The invisible enemies are known, are fought, are sometimes defeated. The crypt is ending and continuation, silence and ongoing speech. The resting place of a man whose work never rests. The tomb marks not ending, but eternal beginning. Beginning of understanding that continues to grow, of battles that continue to be fought, of victories that continue to be won. Conclusion, where light falls on the invisible. The story of Louis Pasteur closes, but the story of humanity's battle against germs continues. Tonight, we have walked beside a tanner's son from the Jura Mountains through laboratories where crystals turned toward light, through fermentation cellars where tiny organisms performed ancient alchemy, through hospital wards where antisepsis prevented infections that had seemed inevitable, through fields where vaccinated sheep survived while unvaccinated sheep died. We have witnessed the moment when a 9-year-old boy received injections that saved him from certain death, have contemplated the institute that preserves Pasteur's legacy, have stood in the crypt where his remains await the resurrection he believed in but could not prove. What have we learned? That the invisible world teems with life, organisms too small for human sight, performing transformations we never suspected, causing diseases we could not explain, inhabiting every surface and every fluid and every breath we take. That these organisms can be identified, can be understood, can be controlled through knowledge rather than through superstition. That vaccination can teach bodies to resist enemies they have not yet encountered. That antisepsis can prevent infections that seemed inevitable. That the ancient plagues need not remain mysterious terrors, but can become challenges that human intelligence can address. We have learned that scientific progress is human progress, achieved by humans with human limitations, human failings, human costs. Pasteur was not saint, was not superhuman, was man who made mistakes and claimed credit he did not always deserve, and held prejudices that his era shared and ours has partially overcome. The controversies about his methods are real. The questions about his ethics have merit, but the achievements are also real. The lives saved cannot be uncounted. The suffering prevented cannot be restored. We have learned that the battle never ends. New enemies emerge, viruses that did not exist in Pasteur's time, bacteria that resist the antibiotics we developed, pathogens that evolve faster than our treatments can follow. The invisible world is not defeated territory, is not conquered kingdom, is not enemy that surrenders. It is constant challenge, constant threat, constant reminder that complexity creates vulnerability. The war Pasteur declared continues in laboratories and hospitals around the world will continue as long as humans exist, will continue as long as invisible enemies threaten visible lives. As sleep approaches, as consciousness dims toward dreams, we carry these lessons into the night, carry the knowledge that the air we breathe contains countless invisible organisms, carry the gratitude that vaccines protect us from diseases that killed our ancestors, carry the awareness that science is ongoing effort, never complete, always advancing, always subject to revision and improvement. Carry the hope that the intelligence that revealed the invisible world can continue to fight the enemies that world contains. The invisible world waits, has always waited, will always wait. But we no longer wait ignorant of its presence, no longer suffer without understanding why, no longer face invisible enemies without weapons to fight them. Louis Pasteur gave us sight where we had been blind, gave us knowledge where we had only fear, gave us hope where death had seemed inevitable. Sleep well tonight. Sleep protected by the vaccines that guard you, by the antiseptic practices that keep your world clean, by the understanding that Pasteur and his successors won through decades of patient work. Sleep knowing that the invisible enemies are known, are fought, are sometimes defeated. Sleep knowing that the battle continues, that others keep watch while you rest, that the cathedral of microscopes never closes, never stops its worship of truth, never ceases its war against the invisible. The tanner's son from the Jura Mountains sleeps now in his Byzantine crypt. The crystals that turned toward light are museum pieces now. The swan neck flasks preserve their sterile silence. The anthrax fields are ordinary farmland now, but the legacy lives. The institute stands. The work continues. Good night. Let the invisible world be invisible for now. Let sleep come as it comes, naturally, gently, carrying you toward dreams where the battles are won and the enemies are peaceful and the microscopes show only beauty. The invisible world. The man who learned to see it, the war that never ends, forever shaping how we live, forever protecting the lives we treasure, forever the gift of a tanner's son who became humanity's defender against enemies too small to see. Sleep now, and wake remembering.